second speaker today is the second Dan David awardee, and this is Professor Gary Ruvkun, who is Professor of Genetics at the Department of Molecular Biology in the Massachusetts General Hospital and Harvard University. And again, simply quoting from the Dan David book, Professor Ruvkun is a key figure in defining the genetic basis for human health during aging, with the discovery of a core set of hormonal signals and signaling pathways that regulate aging and lifespan in animal models, which are likely to act in humans as well. Most notably, he defined an insulin signaling pathway that regulates aging in the C. elegans worm and showed that the essential elements of this pathway are conserved in mice and humans. His work has had an enormous impact on aging research relevant to longevity and later life health. Professor Ruvkun has been awarded the Benjamin Franklin Medal, the Albert Lasker Award, and is a member of the American Academy of Arts and Sciences and the National Academy of Sciences of the United States. And Professor Ruvkun will tell us this morning about genetic and genomic analysis of C. elegans longevity pathways. Gary, please.
It's a pleasure to be here. Let's see here. Work? Yes. Great. It's a great honor to be recognized by Israel and to be giving a talk on aging here. The talk I'm going to give today is about a little bit about the insulin pathway, but I won't do repetition on what Cynthia talked about, but try to intersect with what she talked about, and then talk more about how we've been led by our genetics and genomics into how animals sense toxins in their environment and how that engages the aging pathway of worms and I think also of us. And this is the work of 4 people in my lab: Justine Mello, Dave Shore, Sean Curran, and Yan Chi. And this is supposed to represent the photography strips that Mr. Dan David spread all over Israel and the Middle East. I actually went to a mall to try and use a photography machine, and unlike the 12 families that were ahead of me, I couldn't get it to work. So I was ashamed of myself. So I had to fake this one up. So, okay, this you know about. I wanted to reiterate that this DAF2 mutant that Cynthia talked about is causing this developmental arrest in the Dower state. And I wanted to talk about that a little bit because when Cynthia found that the DAF2 and the Age1 mutants were part of this Dower pathway, that caused many of us great concern who were studying aging, but also who were studying Dower. We had a previous concern, which is, are we in a corner of biology that is so dedicated to a worm that it's not interesting? And this is always a tension in biology of studying the specific in one tiny corner of biology. Who cares? And Dower was really at that edge where there was a whole lot of people saying, well, that is a worm-specific quirk of how they sense their environment and go into this little spore state, and why the hell are you doing that? In fact, my department chair, I'm sure, said it to me more than once. Why? I'm studying oncogenes. Why are you studying Dower? And it was hard for me to. I would just say, go away. I don't want to talk about it. Now, so over time, as we started to contemplate it more and read more, reading is good, it turns out chapter 60 of most biology textbooks finally get to Dower. And the reason no one knows about any of this is that nobody ever gets to chapter 60, right? So if you actually, take your time and read these books, they don't talk about dower, they talk about diapause. And diapause is what insects do if you dry them out. It's what frogs do. They surround themselves with mucus and bury themselves in the sand. People know more about the desert than I do, but I'm sure it happens here too. And this is kind of a weird quirk that's at the end of textbooks, like how animals respond to stress. And this gave us a genetic handle on that problem, number one. The other thing I wanted to mention at this point was the reason I wanted to study dower was my intellectual history, where I had come out of bacterial genetics. Cynthia also came out of bacterial genetics. That is the Olympics of genetics, right? Bacterial genetics are the people who really invented, great genetic analysis. And my first day in a C. elegans lab, when I found out they didn't use genetic selections for their genetics, I was like, what have I done? This is just horrible. What are they? Because they do things by screening. You look in a microscope for the 1 in 10,000. That means, you're sorting 1 in 10,000, where a bacterial geneticist says, comes back the next day and you get your 1 in a million and you do your work on it, right? You let the selections do it for you. So dower was a way to bring selections to what I was doing. It was taking the lazy approach from bacterial genetics and trying to bring it to the field of animal biology, because these dowers are resistant. You can put them in 1% SDS. If you put this animal in 1% SDS, they drink in the detergent, they dissolve themselves, and they're dead in a couple of minutes. The dowers, you come back the next day and they're swimming around. They're perfectly happy because they're evolved to be resistant to that. So that allowed us to do genetic selections. That's why I wanted to work on it. It wasn't an intellectual reason. It was a habit of mind reason. And it was only sort of 2 years later that I said, they were actually kind of interesting, And that was because of the work of Jim Thomas, who really put together this kind of a genetic pathway. And what Jim Thomas brought to the problem was a yeast geneticist's sophistication on how to assign a pathway by looking at phenotypes and doing epistasis. And I won't take you through all of this. I sort of want to gloat about it. This is as complicated a genetic pathway as anybody studies in any organism. And this was constructed mostly by Jim Thomas. We did a little of it in the DAF2 pathway, but it was mostly his lab that put this together by making double mutants between the class that are constitutive for dower and the class that are defective for dower. This is genetic heaven, right? When you get opposite phenotypes and can combine them in pairwise combinations, you can be busy for a couple of years asking, when I make a double, who wins? And you build a pathway by this. And this pathway was constructed based on pure genetic criteria, right? There were no molecules decorating this pathway. We didn't know anything about what their identity was. But it was clear that this was somehow how the animal knows what's going on outside of itself, transduces those signals, and then controls. In the case at this point, we were studying whether they arrested this dower stage or not. Again, the way we think about this is that an animal that's constitutive for dower is hallucinating bad conditions. And an animal that's defective for dower is hallucinating good conditions. Again, the use of the term hallucination comes from my background of growing up in the 60s, right, where we can actually visualize these sorts of things. It pays. Being in the diaspora museum like this, I believe in moving through cultures. And I think one of the great things about modern biology is how people do postdocs in different fields from what they did their graduate work in. And that feeling of being totally uncomfortable being an immigrant into a new culture that you're studying is absolutely key to discovery and invention. So So when Cynthia's lab discovered that age1, which was the first aging gene discovered by Michael Klass and Tom Johnson years ago, and DAF2, which was the second aging gene discovered in Cynthia's lab, that the two of them were very potent at regulating aging. And then when she figured out that they are actually acting in the same pathway, that was an amazing revelation, because there was a sense that aging was going to be extremely complicated. Not just that a single gene mutation might affect it, but that there might be hundreds of different pathways. And here we had the first two hints on how it was working, and they were acting actually in the same genetic pathway. That was really a revelation that maybe it wasn't going to be a 100-year plan to study this. Maybe you could figure it out in a few years. And it's very important that it intersected this complex genetic pathway that network that had been put into place. So that it allowed all of this pre-existing edifice to be marshaled to the study of aging. And so we could say, okay, does this live longer? Does this live longer? Does that live longer? Who suppresses that? Et cetera. And so, boom, it progressed. Within a year, it sort of clarified itself that this is the pathway that regulates aging. It didn't look like this one did, although 10 years later, Colleen Murphy has discovered that this other pathway regulates aging. I won't go into these other pathways much. These genes turn out to be TGF-beta endocrine signals. It's an analogous set of endocrine signals to the insulins, probably working in parallel signals to regulate Gower. And then this turns out to all be insulin signaling. And then there's receptors for environmental inputs. So when we figured out that the DAF2 corresponded to the mammalian insulin receptor, the way we figured that out was by simple genome comparisons, where we took the protein sequence of the worm gene, which is about 1,000 amino acids long, and sent that data to, this was 1997, so the human genome wasn't yet finished, but it was like 20% done. And of course there was all the previous protein and DNA sequence work that people had done on a scattershot sort of basis. And it was obvious instantly that this was the homolog, because it has very good homology both in the ligand binding domain as well as in the protein kinase domain. Okay, who's got their... Turn off your phone. Everybody, turn off your phone. Zero tolerance here. So it's got homology in the ligand binding domain as well as in the tyrosine kinase domain. Now, this was important for two reasons. It was, of course, important for us that we understood we were working on an endocrine pathway. And so we could engage in incredibly rich literature on human insulin signaling, because, of course, the first molecular medicine was on insulin. So it's an incredibly highly evolved field. And we could see what had been learned about how that receptor works, who it couples to, et cetera. It was a bit of a one-way street, though, because the endocrinologists were not as sophisticated as we were. Even though endocrinology is kind of the priesthood of medicine, I think, they were like, well, worms, we're doctors, who are you? Okay, so this is where humans are in the evolutionary tree, and here are the worms, and this is the late 90s, and they're not having any of this, right? That the worm can teach them something about how insulin works. Give me a break, you guys, you study your little Dowers, and come back to the big leagues when you're ready. So we had it to ourselves for many years. Now, over time, the endocrinologists have realized that we have stuff to teach them. So, for example, out of the worm genetics came that the insulin receptor couples to a PI3 kinase, and that couples to a set of other kinases called PDK and AKT, and that couples to a transcription factor called FOXO, as you've heard from Cynthia. And the point I want to make is that you have to get out of your little ghetto, right? So the mammalian endocrinologists were very few of them were listening to this, but there were a few who did, and started to realize that the transcriptional outputs of the insulin receptor were actually important for how you think about diabetes and how you think about how IGF-1 signaling works. And one of the most satisfying things to me, and I'm sure it is to Cynthia too, is that the FOXO output of the insulin receptor, which was not a field in diabetes research, is now a major field in diabetes research. And if you go to meetings, 3,000 people who work on diabetes, a quarter of the sessions are about the transcriptional outputs that come from FOXO, and that comes directly out of the worm research. And that's extremely satisfying. It has nothing to do with aging as a central component, but it's influential. The other thing to realize is that this was one of the first my first experience of genomics fusing fields, right? That by virtue of what comes out of a blast analysis when you compare a protein sequence, we were working in the insulin signaling field, and of course had that literature to engage, and they had our literature to engage. And what that means is you have to think about biology as something like a 10,000 gene project. That there's really only 10,000 genes out there, period. That all the animals are sharing those 10,000 genes, and we have to divide that up between fields. Or another way to think about it is, if you're working in insulin signaling, your competitors are not just the people who work in insulin signaling, it's the people who work in mosquito insulin, the people who work in worm insulin, et cetera. So that one has to read more broadly than one did a decade ago, or one gets to read more broadly than one did a decade ago. So this, when we found that the insulin receptor was involved, we thought, well, maybe it's related to caloric restriction, that of course insulin has everything to do with feeding in humans, and so maybe this is the homologous state of what happens in caloric restriction in animals. So what do we know about animals so far? And this is work from Bev Pagan. This is not our work. And what she has done at the Jackson Labs in Maine is to look at mice. And these are the lifespan of various mouse strains. And you can see the lifespans vary from 250 days to 900 days. It's a huge span of lifespans. And this is all comes out of the mouse collectors of the 19th century, right? The mouse strains that are used in research were the pets of the Victorian households in the 19th century. And when she did QTL analysis to ask what explains the variation in lifespan, one of the major quantitative trait loci that she mapped is right in the insulin receptor locus, the IGF-1 receptor locus, which is what the worm genetics was saying. So what this points to is this another tension that one always finds, especially from reviewers, that what you're claiming is very naive. The idea that if the IGF-1 receptor has some function in the worm, in this Dower arrest, well, what would Dower have to do with a mammalian lifespan? That's very naive. And the key to invention in science is to go with naive. Don't worry about what your reviewers say. Your reviewers will always tell you you're naive, and they're wrong. They are wrong. That's why they're reviewers. So, the fact that there's a hormonal pathway in lifespan, does that sort of mesh in any way with what we know about lifespan? And Cynthia mentioned how much lifespan varies. So these are the lifespans of a whole variety of animals, and you can see that it's highly varied in the animal lineage. And hormones are the kind of thing that do vary over fast evolutionary time. So for example, the reason Scandinavians are so tall is that their growth hormones have a higher set point than the rest of the world. There's been a selection in the northern latitudes to get bigger, probably to have a higher surface area, so you D or A or whatever it is, the light one. It has a bigger antenna. So the idea that there's a hormonal pathway involved in aging makes a lot of sense. It does not fit with what the evolutionary theorists were telling us we should have been thinking. But that's another sort of rule that I live by, which is ignore what evolutionary theorists tell you and let the genetics sort of rule the game, just the variation you can find. So this is about a decade ago, 15 years ago, and then the question is, so we knew something about aging, but it wasn't done in a comprehensive fashion. It was, let's see if we can see some aging genes, and it was sort of the first dipping the toe in the water, finding that the insulin pathway, and so the next level of it is to do a more comprehensive screen, and worm genetics is good for that because of the discovery of RNAi and the development of RNAi libraries. So the way RNAi works is actually quite miraculous, that you can put double-stranded RNA into an organism, and there's a bunch of enzymes that I'm blocking out here that will chop up that double-stranded RNA into 22 long RNAs that will then find complementarity, find a target mRNA, and then destroy that mRNA so its levels go down, and that leads to a decrease in the amount of target protein. And so you can then make libraries that have each double-stranded RNA from every worm gene. So you tile along the chromosome. Every chromosome has, in the case of the worm, about 3,000 genes. So you can just, from the genome sequence, develop RNAi libraries, and that was done by the Erringer lab. And these libraries were available to the worm community in microtiter wells. So in a 24-well plate, you can grow 100 worms in here and feed the worms double-stranded RNA that are in an E. coli strain that worms eat, and it will inactivate genes by feeding. This is even more miraculous than RNAi, the fact that you can feed a worm something that will inactivate its gene. It's totally amazing that this worked. This was invented in Andy Fire's lab by Lisa Timmons. If anybody had proposed to inactivate genes by eating, that's a definition of failing a qualifying exam. But it worked. And so we do it. Now, when I say it's miraculous, it works, it sort of works 75% of the time. What does it mean 75% of the time? We know a lot about worm genes from the history of worm genetics. So there's something like 1,000 genetic loci that have been well studied, we know a lot about. And so that we can feed those 1,000 loci and ask what percentage do we get the phenotypes we expected. And you get about 75%. What you don't get is the nervous system, or you barely get the nervous system. For unknown reasons, RNAi doesn't work very well in neurons, probably interesting reasons why it doesn't work well in neurons. So it means when I say we've comprehensively studied X, Y, or Z, we're sort of 75% comprehensively. The other thing is these libraries are sort of 94% complete. Nobody's ever gone back and finished the 6% because everybody's too lazy compared to the Erringer lab. We always have a joke in the lab that if we worked on that 6%, it would be our 6%. Nobody else would work on it, but nobody does it. So I'm going to tell you about one of the screens we've done in the last couple of years. And this is, here there's 20,000 genes that you can feed the worm and ask who increases lifespan. In this particular study, we didn't look at all the genes. We only looked at the genes that when you feed it to starting from an embryo state and ask what happens, there's sort of hundreds of gene inactivations, hundreds of different gene inactivations that will cause the animal to arrest at the larval stage 1, and a different set of genes will cause them to arrest at larval stage 2, etc. So there's something like 3,000 gene inactivations that sort of make the animal less healthy. And the initial way anybody would think about these who does genetics is that these are the essential genes. These are the components that are you can't inactivate them and be alive. Things like the ribosome, the cytoskeleton, really core components of cells. But what we did was RNAi has this lovely capacity where you can say, okay, maybe these are things that are needed to grow up, but what if we inactivate them late in life? So we can still ask, do they affect the lifespan by beginning to inactivate them sort of after all the early stuff has happened? And we got a lot of hits out of that. Then you can do this, take the hits out of that and go back and say, well, now what happens if we actually start to inactivate them much earlier? And what we find is that many of those hits that came out of this screen also cause an arrested larvae phenotype, but those arrested larvae actually live longer, right? So they're not dying. It's not that they're essential genes. And the way to think about it is that maybe this arrest point is a program, just like Dower is a program. There's a response here. So here's what some of the lifespan curves look like. So here's our control strain under the conditions that we do the RNAi. They're living sort of 3 weeks. And then these are protein synthesis genes, things involved in initiation factors for translation that are not as good as DAF2. So here's DAF2 out here by RNAi, about twice as long. But really the next best taxon rank of lifespan regulators. So if you go through the whole genome and ask who's the most robust at regulating lifespan, the insulin pathway wins by a lot. But the next rank are these guys. And if you go through, this is sort of in rank order of who does the lifespan the most after the insulin pathway, and these are things involved in translation initiation, or sort of the top three, lots of RNA binding proteins. Here, this is a mitochondrial component. This is the factor that actually generates ATP, not part of endocrinology at all. None of this is part of insulin pathway endocrinology. Sorry. So, one of the things we notice, this is very dark, maybe you can see it, is that these gene inactivations that cause developmental arrest, here's a control. This is showing maybe a centimeter on a plate. These are bacteria, and the little squiggles in the bacteria are the tracks of worms. And each of these lines here is a worm magnified. If you give a control RNAi, they're almost all on the lawn. But when we give them these RNAis that cause developmental arrest, you can see many more animals move off the lawn. Here you can especially see that these guys are moving away. So these guys know that these are toxic gene inactivations. They're not feeling very well, and they're trying to get away from that. And yet they're living longer. So we decided to explore this a little bit, and I'll show you sort of the difference between, on the right side is a gene inactivation that leads to an increase in longevity, but it also causes, this is causing larval arrest, because this is starting the feeding at L1. This is the control, and each of these colors here is what track it will take. So that's a tracking software that sort of tracks the animals. So what you can see are animals on the control are quite happy. They're like grazing cows. They're just chewing their cud. They're eating the E. coli. They feel fine. These other guys are a little smaller and thinner because they're arrested animals, and their tracks tend to be much faster. These are filmed at the same rate. This is 20 times sped up. But their tracks are much faster, and they don't tend to turn as much. And you can actually plot this out. And so you can say the mean speed of an animal on a control is about half to a quarter of what it is on these toxic RNAis. And my favorite statistic we've ever had in the lab is called tortuosity, which I think is fun. And that's a measure of how often the animals turn. So on a benign source, they constantly change direction. But on a source that's toxic to these guys, that causes an increase in lifespan, but it causes an arrest, they don't turn nearly as much. And it's basically a measure of how they tend to move in long trajectories. And this is a lot like bacterial chemotaxis, where an animal changes whether it tumbles versus moves in straight lines. This is a way to get away from where you are. So Justine went and did about 6,000 gene screen for what are all the gene inactivations that will cause this leaving of a lawn. And she classifies it based on where they arrest. So if they arrest at L1 or L2, etc. And you can see that some of those inactivations cause 40% of the animals to leave a lawn, even 50% at L2, and even more later. So she got about 400 hits that cause the animals to want to leave a lawn. Many of these increase lifespan, but not all of them, and we haven't yet figured out the intersection of lifespan. This is too many to do lifespans on. So if we just look at who comes out of this screen, we get 55 out of the 76 subunits of the ribosome. Almost everything comes out. We get much of the proteasome, much of the mitochondria, very conserved vacuolar ATPases. The molting genes are actually very poorly conserved, but we know a lot about them because we did a screen for full genome screen for RNAis that affect molting, etc. We can even take these gene inactivations and we can inactivate them in different tissues. So there's ways to get rid of the RNAi machinery by having a mutation that is incapable of doing RNAi, and then adding back that RNAi machinery to either the muscles or the intestine or the hypodermis. And for example, if we inactivate the proteasome, our control, 25% will leave. But if we inactivate it, sorry, I'm colorblind, so I cannot tell you what these scores are. So you can tell me. In one tissue or another, it will cause it to leave, whereas other tissues it won't. So RNAi in particular tissues will cause an animal to leave a lawn. And what that tells you is that you're activating an endocrine system, that there's some surveillance going on in particular tissues that then spreads to the rest of the animal to then cause a behavioral change. And so, for example, here, where we're inactivating the HSP6 gene, you can see all these animals off the lawn. And even if we only inactivate it in the gonad, these animals are off the lawn. So there's a signal saying, my HSP6 is not working, I'm not happy, and even if it's only in the germline, it sends it to the rest of the animal. So we know that DAF16 is key for low insulin signaling. Cynthia showed you that. The question is DAF16 key for these other responses? So we know that DAF16 is the key transcription factor. What happens to this leaving response in a DAF16 or DAF18 mutant? And so, for example, here's a case where the DAF18 mutant really strongly suppresses inactivation of a ribosomal gene. DAF18 is quite similar to DAF16 in the sense that it's the PTEN homolog that's involved in the signal transduction. It's Dower defective. It takes care of both the transcriptional outputs as well as the kinase output. So it's sort of higher on the pathway, regulating more of the outputs in this signaling cascade. And it does better at suppressing than DAF16. So, for example, here's a case where DAF16 does not suppress, but DAF18 does. So that what that says is that this pathway that is involved in sensing a toxic environment works via insulin, works via the insulin signaling cascade. So part of how the animals feel is due to dysregulation of their insulin pathway, which is related to the lifespan phenotypes. So what do we know about the genes that are the targets of this? And what we've been doing now is we can take every worm gene we've looked at, we've asked what's the most conserved worm gene in humans. And so we do a rank order. And this is actually work, a much larger project that Yuval Tabak is doing, who's a postdoc in my lab, who actually trained here at Tel Aviv University. And one aspect of what he's been doing is asking, okay, what are the most conserved proteins on a per, normalized to length between all organisms, and doing human worms for obvious reasons. This is the rank order. So the most conserved is calmodulin, and then histone H3, actin, etc. And it turns out that of the top hits we get in our aversion screen, the actual wanting to get away from the gene inactivation, they tend to be very, very highly conserved proteins. And that was the first hint of what might be going on here. So if we ask, here's the rank order of genes. This is the top 5 lists, right? And so this is the most conserved, next. Which are hits in our list? Well, calmodulin comes out big time, actin comes out, histones come out, etc. So if you ask, what's the average conservation of C. elegans genes? It scores about 0.1 in our ranking. In our gene inactivation library, we're asking for essential genes. They're about twice as conserved as what you would get on average expectation. And the genes that cause aversion are about double the conservation of the library. So it's very highly enriched for conserved genes. So why are the genes that we seem to get out of this screen the most conserved genes? And the hint for this came as we started to sort of read. We would take our top hits and say, well, okay, what's known about this? And as you'd read abstracts, because nobody ever reads the papers, you just read the abstracts and say, oh, well, okay, next. And you go through a couple hundred of these and you go, they mention natural products targeting this gene a lot in abstracts. I'm seeing natural products over and over and over. So, for example, the ribosome comes out, and the ribosome is a huge target for antibiotics, for drugs that are meant to inhibit other organisms. And so, things like erythromycin in bacteria, but there are antibiotics that affect eukaryotes, like hygromycin. So these are naturally evolved toxins that are targeting highly conserved proteins. Now, let me give you one other example. So here's ATP synthase, right, which sticks through the membrane and does the proton gradient to then generate ATP with a kind of a motor, like a flagellar motor. And it has a natural product that inactivates it called oligomycin. And it was the fact that these very conserved proteins have natural products that was kind of the hint of what's going on. So these conserved proteins tend to form structures and components that are sort of unique in various clades. For example, bacteria don't have a Golgi. They don't have a nucleus. Everybody's got ribosomes, and there is a war going on out there, right? There are organisms that make drugs that are trying to target conserved elements. And why would you want to target conserved elements? Well, that makes your drug work on everybody else. If you target something unconserved, you're only targeting one organism. If you target very conserved proteins, you're targeting a whole taxonomic universe. So the prediction of this is if we used drugs, we should get the same behavioral response as if we do gene inactivation. So, for example, we can take a mitochondrial drug, antimycin, and 2 hours after giving it to a lawn of benign bacteria, so these are not inactivating any genes, these are the control bacteria, we drop on the drug and the worms instantly sense it and try to get away. So they have an instinct, which is if they sense a drug, they don't want to eat. Well, that's an instinct we have too, right? If you feel rotten, you don't want to eat. So I call this now the Nausea Project. We are studying the genetic basis of feeling awful, right? And our worms, you know exactly how these guys feel, right? You know exactly how they feel. And it's a feeling of being poisoned that our common ancestors a billion years ago had because they were poisoned. And there is a way to deal with that. So what do we think is going on? So what does that say? Sam, but I don't know what that was not on mine. This is probably who I ripped off this image from on the web. A little time bomb that goes off. So what we think is that there's a surveillance program, that the ribosome, the integrity of the ribosome, for example, is being monitored. And that when we do a gene inactivation of that ribosome, or when we put in a drug that inactivates the ribosome, that's sensed. We don't have any idea how it's surveilled. It could be the production of some sentinel peptide that gets constantly assessed, and if you don't make that peptide, you know you're in a poison. But it's a dedicated process of knowing that you're in a poison that will then cause the expression of some hormone, because we can inactivate these genes in one cell type and cause the animals to try to get away from the bacteria. But also probably inactivate a xenobiotic detoxification. So I think we're also doing toxicology here. So remember, we started studying aging, and now we're moving into the endocrinology of nausea and also into toxicology, but they're all connected. So what we think is that translation, is, just as an example, is something that's targeted by antibiotics. Those antibiotics are produced by living organisms, and so when translation is not working quite right, the animal instantly thinks it's in a poison. So when we do RNAi of translation factors, the animal has an instinct, a calculus that says, oh, I'm in a toxic environment, and so I'm going to developmentally arrest because there are my enemies out here that are producing chemical toxins that are trying to kill me, so I will definitely not reproduce. That would be a big mistake. So instantly suspend reproduction, but also stop the aging process, because you are not in a fast track, produce a big brood. You're in a slow track, get the hell out of there, and save all your resources to produce a brood somewhere else. So what do we know about these poisons that are natural? So rapamycin, Cynthia mentioned, so that's produced, anything that says mycin, that means it's produced by some organism, right? Erythromycin is produced by streptomyces. And these have been studied for many years. When antibiotics were first discovered, it was, how are they made? You know, the organic chemists really care about how these things get put together. And they were studied genetically because these are made by a bacteria. Bacterial geneticists look for mutants in the host bacteria that no longer make the drug. And what they found when they did that was that there is a biosynthetic machinery that makes these big honking proteins. They're called polyketide synthases. These are sort of 8,000 amino acids long. These are, 10 times as big as what we're used to thinking about. And they have these multiple cassettes that take 2 carbon units and condense them together and put out multiple carbon units, decorating them with hydroxyls, and they're like organic chemists, but not as arrogant. And they spit out these polycyclic compounds that are toxic to organisms. Now you can recognize these in genome sequences, so you can look for polyketide synthases in any environment you choose to look in. And that is the secondary metabolism of microbes. If you look in any one genome sequence, like here's the yeast Aspergillus, there's 27 different polyketide synthases, and 19, another class is called the Nonribosomal peptide synthesis. These are also making antibiotics. So just this one organism makes 50 different toxins that are all about their ecological life. It's all about trying to antagonize who they encounter in the soil for essentially eating their lunch. And so with genomics, there are thousands of these that are now going to be available. In my mind, when anybody talks about chemical screening, these are the things I want to screen, because these are the chemicals that have been through evolutionary selections. These are the things that do enough that they're there, right? The chemicals that chemists synthesize, they have not been through that evolutionary sieving process. There's too many possibilities. So this is really all about, so this is us in the tree of life, and animals are all in one branch because we're really not very different from each other. Even our worms are hardly discernible. And it's all the rest of phylogeny. Many of these bear polyketide synthases, and they are trying to do us in. Okay, so I've told you that. So how are we trying to work out how the animal detects poisons? And I'll bring you back at the end to how it might be related to aging. So the way we do this is that there are green fluorescent protein fusions to these various poison detection pathways. So this is an endoplasmic reticulum unfolded protein response. If we give the animal a natural product that inactivates the ER, in a couple of hours it turns on this HSP4. This is a green fluorescent protein for the mitochondrial unfolded protein response. If we give it this mitochondrial toxin, it comes on, etc. So we have all these different stresses that we can give. We try to work with things that are natural products, but not exclusively, like sodium azide is a very simple molecule that's sort of not made by a PKS. And then if we go through our gene inactivations that increase lifespan, so these are the lifespan regulating gene inactivations, many of them activate these xenobiotic response elements. So when we're inactivating these guys and therefore increasing lifespan, perhaps what's playing into the increase in lifespan is this xenobiotic response pathway is getting kicked in. So xenobiotics toxicology is a big field, right? A lot of people care about what response to drugs, especially the drug industry. The pharmacokinetics of a drug, what happens when you take a pill, is all about the detoxification, how the drug is sensed, and then how it's decorated with basically hydroxyls and sulfates and UDP glycosylations to give it more and more and more handles for your kidney to then sieve it out and you pee it out. And that's the pharmacokinetics of drugs. And it's a very mature field at the level of description. So these are all the various drugs, gene proteins that get induced that are decorating medicines when you take medicines. What's totally unknown, or almost totally unknown, is what are the gene regulatory cascades that induce these? How does the animal know that a drug is in the system? And there's a little bit of literature on this, but it's involving nuclear hormone receptors, and my guess is that it's barking up the wrong tree, and that the worm genetics is going to help really figure out how the genome is surveilled. Now, one hint to how this might work comes again from Jim Thomas. So, he's one of the heroes in my worldview. He lost interest in much of the Dower pathway and is now just a genomic savant who stares at genomes and thinks about it. And what he noticed when he looked in the worm genome, so these are each of the worm chromosomes, he noticed these clusters. For example, this is a bunch of cytochromes that are involved in putting a hydroxyl group on drugs and poisons. And there's a whole cluster of these cytochromes. So they're all one gene right next to another. And when you see a cluster of genes like that, in my lab, you think instantly RNAi heterochromatin, sort of regulation by a chromosome structure. And we have a handle on that because the other half of the lab works on RNAi pathways that are involved in how the small RNAs sort of engage chromatin regulation. And so we have these sub-libraries that we've made from our other screens in the lab. And so we could feed the animals those sub-libraries and ask whether any of the sub-libraries involved in RNAi make the animals poison blind. We're looking for animals that when we now give them a poison, they don't turn on GFP. And out of this sub-library, for example, here's a gene inactivation that sort of turns it on about 1/8 the amount that we get from a wild-type animal. So when we give the animal tunicamycin, this gene inactivation failed to turn it on. This came out of a library that aged faster. This hit came out of a library that's involved with microRNA function. This came out of a library that was involved with RNAi. So the reason we think micros could be involved is that those regulate translational control off a ribosome. So if a microRNA is inhibiting a target on the ribosome and you inhibit the micro pathway, you can rather instantaneously produce the protein. So it gives you a fast dynamic response, which was the reason to screen that. But again, we're just at the level of sort of pulling these things out now. The other approach is to actually go for drug resistance. So if we take a wild-type animal and treat it with these natural products, we can get an arrest, very strong arrest. And now we can do an RNAi screen for guys that don't arrest. And when we do that, we get a whole bunch of genes that come out of these screens that came out of our RNAi libraries. And these are things that grow up on hygromycin. They're resistant to hygromycin, probably because they're constitutively expressing all this drug detox pathway. So notice that our interest in drug resistance is completely perpendicular to the way most people think about drug resistance. Most people, they say, I want something specific for my one drug, or else it's some kind of uptake thing. I'm not interested. We're interested in general pathways of toxin resistance. We're not so interested in sort of how you mutate the binding site for a drug. And so now we're taking these poisoned line mutants and going back and asking what is the lifespan phenotypes, but we don't have that data yet. Okay, so I better finish. I think I'm done. Just one last point I want to mention is that what we're dealing with here is a system of how you detect poisons and how you detoxify it. And we expect that to be very different in females and males. And one reason, the reason for that in mammals is that females cannot take a bout of a toxin the way a male can. If it's a teratogen and you're 2 months pregnant, this is not good, right? A male can puke and go drink beer the next day, but a female who's pregnant cannot. So the prediction is that this pathway should be much more sensitive in a female than a male, and that maybe inappropriate activation of this pathway is something, maybe explains the gender bias of something like anorexia, which is sort of 10 to 1 females. And I know that, I debate this constantly. It's all Vogue magazine. Well, I don't believe Vogue can give you a 10 to 1. I think it's biology will give you a 10 to 1. I'll let Vogue give you a 2 to 1, but not 10. The other thing to think about, though, is the long, you go to a nursing home, it's all women, right, at age 90. And why is that? Well, again, maybe the fact that there's so many toxins in everyday life that activate this pathway in females more than in males, maybe that explains the longevity. And maybe it's not about the males having horrible, sneaking hot dogs and, working in chemical factories. It could be real simple biology. So we're looking now. So we're started a project to look in females who have anorexia, whether they've upregulated their xenobiotic detox pathway. We're just starting that. So anyway, I'll end there. Thank you very much.